affects reptiles with temperature sex determination to a degree that is not yet well understood. The rising temperature due to human impact directs a need for a better understanding of how changes in the temperature of nesting environments may dictate the survivorship and fitness of the species. The objective of this research was to reveal the effects incubation temperatures had on the performance of hatchlings of the loggerhead sea turtle. The aim was to determine what significant developmental effects would arise as the temperature was differed within the nests. This research is becoming extensively important as the rate of climate change is increasing. The fear that the turtles may not be able to adapt to the rising temperatures in the nesting area is evident as this threatens their survival as a species. As juvenile and hatchling ratios are typically female biased, if the future climate increase doesn't rise to a fatal degree, the fear that the population may be feminized is also present among researchers, supported by the study of breeding sex ratios in adult leatherback turtles may compensate for female biased hatchlings by Kelly Stewart and Peter Dunn. The artificial incubation process of the eggs was conducted in the Centre for Marine Sciences and Technology, using a mechanical incubator so that temperatures could be monitored and kept constant. Data was collected from both the year 2011 and 2012. These experiments tested 68 hatchlings in 2011 and 31 in 2012 that hatched from incubators with 11 different constant temperatures ranging from 27 degrees to 33 degrees. Several tests were taken and then data was recorded from this. These tests included a riding response, crawling speed, and a 24-hour long swim test. Results led to the conclusion that hatchlings incubated at 27 degrees showed slower locomotion activity and abilities. Also, no hatchlings survived to be tested at or above 32 degrees.
The graph shown represents the percent of hatched eggs, dark grey, and the percentage of hatchlings that survived, light grey, in 2011 overall. The maximum number of hatchlings and also survivors can be seen highest at 29 degrees and minimum seen at both ends of the temperature extremes.
Tests taken of loggerhead sea turtles show that the optimal range for performance after hatching is of a mid-range temperature. This is supported by previous studies such as the climate-driven egg and hatching mortality threatens survival of the eastern Pacific leatherback turtles, conducted on the beaches of Australia and North Caledonia, where a noticeable decrease in locomotion ability in warmer extreme is present. Throughout the test, the temperature of incubators were taken and recorded daily, making sure that there was no differences in temperature. No shelf that was used in the study differed by more than 0.5 degrees during the incubation period. The final experimental groups for 2011 were 27 to 32.5 degrees and 26.5 to 33 degrees for 2012. In conclusion, controlled laboratory experiments and tests supported previous studies undertaken that incubation temperatures affect the performance of loggerhead sea turtle hatchlings and therefore lifespans. Generally, turtles incubated at a lower end temperature extremes saw a significant decrease in locomotor ability, and turtles incubated at higher temperatures rarely survive. Further studies need to be conducted into the incubation temperatures of turtle nests to determine whether effective climate change over the next 100 years forces the turtles to extinction, or if adaptation may be able to occur to the fluctuating degree of the nest environments.